Good morning to everyone. Today we will discuss about serum protein electrophoresis. Two commonly used separation techniques in clinical biochemistry. One is electrophoresis and second one is chromatography. Today we will discuss electrophoresis. This technique was invented by Arne Thyselius. He got Nobel Prize in 1948. Next, coming to the principle, electrophoresis is defined as movement of charged particles through an electrolyte when subjected to an electric field. Positively charged particles move to cathode and negatively charged particles move to cathode. Here, the migration of charged particles under the influence of electrical field towards opposite charged electrodes. That means cations move to cathode, anions move to anode. It is mainly used for separation of closely related compounds from mixture like proteins, isoenzymes, lipoproteins, hemoglobins, immunoglobulins and many other compounds can be separated by electrophoresis. This is the principle of electrophoresis. Next, coming to the different types of electrophoresis. Electrophoresis is classified into different types. One is moving boundary electrophoresis, zone electrophoresis, immunoelectrophoresis. In zone electrophoresis, paper, cellulose acetate and gel electrophoresis. You can observe here moving boundary electrophoresis or capillary electrophoresis. Under the zone electrophoresis, three types: paper electrophoresis, cellulose acetate electrophoresis, gel electrophoresis. In gel electrophoresis, two types: agarose gel and polyacrylamide gel electrophoresis. That is the page. Polyacrylamide gel electrophoresis, immunoelectrophoresis, isoelectric focusing electrophoresis. Next, coming to the factors affecting electrophoresis. Electrophoresis depends on the different factors. One is net charge on the molecules. It's depending on isoelectrical pH of proteins and depends on the pH of buffer and supporting medium. Supporting mediums are paper, gel, cellulose acetate supporting mediums, and it depends on the strength of electric field and size and shape of the molecules, and it depends on the temperature of the buffer. These are the factors affecting electrophoresis. Next, coming to the clinical applications of electrophoresis. It's mainly used for separation of plasma proteins, serum proteins in case of nephrotic syndrome, cirrhosis of liver, chronic infections, alpha-1 antitrypsin deficiency, etc. It's used for separation of isoenzymes, that is lactate dehydrogenase, creatine phosphokinase, and used for separation of nucleic acids and separation of hemoglobins and its variants in case of thalassemia and sickle cell anemia. It's used for separation of lipoproteins. Lipoproteins are HDL, LDL, VLDL, etc. and separation of immunoglobulins in case of chronic infections, and it's used for identification of monoclonal antibodies in case of multiple myeloma, and it's used in blotting techniques, southern blot, northern blot and western blot techniques. These are the clinical applications of electrophoresis. And this is the electrophoresis apparatus. You can observe here, this is the electric connections, and this is the agarose gel slide, and these two are filter papers so to supply the buffer, and this is the tray with buffer. This is the electrophoresis apparatus. Next, coming to the separation of serum proteins by agarose gel electrophoresis. What are the requirements for serum protein electrophoresis? First is electrophoresis apparatus with power pack, microscopic slides, tris buffer, pH is 8.6, agarose powder, Wattsman number 1 filter paper for wicks, and bromophenol blue, it's used as tracking dye, and 70% methanol, it's used as fixating solution, and amido black staining solution, and 70% acetic acid is used for destaining the slide. These are the reagents required for serum protein electrophoresis. Next, coming to the stains. To visualize and locate separated analytes, these are the different stains it's used to identify the different analytes. First is serum proteins. We already discussed, used bromophenol blue or amido block. Then DNA. For DNA, ethidium bromide cyber green cyber glowed. Then for RNA, acridine orange. For lipoproteins, Sudan block oil O red, fat red 7B stains. Isoenzymes, nitrotetrazoleum blue. For CSF proteins, silver nitrate stain. These are the different stains used to different analytes. Next, coming to the procedure. Take 2 clean microscopic slides are coated with thin film of agarose gel. Few microliters of normal and test serum are mixed with tracking dye. Tracking dye is bromophenol blue, which is applied with the help of cover slip at 1/3 of cathodal end of slide. Electrophoretic chamber is filled with buffer. Then agarose gel slide is placed over the support media, and contact is made with buffer through the filter paper wicks. The lid is closed and electrical connections are made, such as 3 milliamperes of current is supplied per each slide. The electrophoresis is run for about 2 hours. The electrophoretic run is stopped when the tracking dye reaches anodal third of slide. After this, stop the electrical connections and slide is taken out, and proteins on the slide are fixed on the slide by immersing the slide in methanol for 10 minutes. Then slide is immersed in staining solution, that is amido block, for 5 minutes. Excess stain is removed by immersing in destaining solution. This destaining solution removes the excess stain. Then the slide is dried with the help of hair dryer or by keeping it in hot air oven for 30 minutes. The bands on the test are compared with normal slide, and the quantitation of proteins in each band is done by using densitometer. Next, coming to the quantitation of proteins. The electrophoretogram can be scanned using densitometer, and each bands are quantitated. Electrophoretic bands are quantitated by using densitometer. In the densitometer, light is passed through the agarose gel plate, then absorption of light will be proportional to the quantity of proteins present on the band. Another method is calorimetric method. The quantitation is by removing the stain from each band by using the calorimetric method. These are the quantitation of proteins. Next, coming to the serum protein electrophoresis bands. Proteins are separated into 5 bands. One is albumin, alpha 1, alpha 2, beta, gamma globulins. Normal serum is separated into 5 bands. Albumin, it's composed of 55 to 65% in total proteins. Alpha 1 globulin 2 to 4%, alpha 2 globulin 6 to 12%, beta globulin 8 to 12%, gamma globulins are 12 to 22%, and major is albumin 55 to 65% in total protein. Next, coming to the serum protein electrophoresis bands, and this is the normal serum protein electrophoresis bands, and this is the plasma protein electrophoresis bands, and this is the point of application. And here anions moves to the anode and cations moves to the cathode, and these are the bands. And albumin is moves faster because albumin is low molecular weight. Then plasma protein electrophoresis, and here if we used plasma sample, fibrinogen band is seen in between beta and gamma globulins. Here plasma is showing fibrinogen. This is the fibrinogen band in between beta and gamma globulins. Next, coming to the major proteins in each band. Alpha 1 globulin composed of alpha 1 antitrypsin, alpha 1 acid glycoprotein, alpha phytoprotein and retinol binding protein. Alpha 2 globulins are composed of hepatoglobulin, alpha 2 microglobulin and ceruloplasmin. Beta 1 globulins are composed of transferrin, hemopexin. Beta 2 globulins are beta 2 microglobulin. Gamma globulins are composed of IgG and C-reactive protein, CRP. These are the major proteins in each band. Next, coming to the electrophoresis pattern in diseased conditions. And this is the normal pattern, and this is the disease condition, and this is multiple myeloma. In multiple myeloma, beta and gamma region in between, M band is seen in between beta and gamma globulins, and this is the multiple myeloma. Next, coming to the chronic infection. In chronic infection, gamma globulin is increased and alpha 1 and alpha 1 and alpha 2 bands are increased. In chronic infection, increase in gamma region and increase in alpha 1 alpha 2 bands. Next, coming to the nephrotic syndrome. In nephrotic syndrome, albumin is decreased and alpha 2 band is prominent, and this is albumin band is decreased, hypoalbuminemia, and alpha 2 band is prominent. Next, coming to the cirrhosis of liver. In cirrhosis of liver, albumin is decreased. These are the electrophoresis in diseased conditions. First one is multiple myeloma, cirrhosis of liver, chronic infections, and this is the electrophoresis apparatus. Next, coming to the polyacrylamide gel electrophoresis. This is the one of the technique to separate the macromolecules of proteins and nucleic acids. The mobility depends on the length because proteins sodium dicyl sulfate complexes have similar mass to charge ratio. It mainly depends on the length of the molecules, and sodium dicyl sulfate polyacrylamide gel electrophoresis is used for separation of proteins. And here sodium dicyl sulfate is used as eliminates the influence of structure and charge for the electrophoresis, and polyacrylamide gel act as supporting media on which the molecules are separated. Sodium dicyl sulfate eliminates the influence of structure, and polyacrylamide gel acts as supporting media. Next, sodium dicyl sulfate polyacrylamide gel electrophoresis is method of separating molecules based on the differences of their molecular weight. The SDS molecules are negatively charged and binds to the proteins. One molecule of SDS for every 2 amino acids. By binding to the proteins, sodium dicyl sulfate destroys their structures, secondary, tertiary and quaternary structure. Then it converts into negatively charged linear polypeptide chains. The proteins converted by SDS are negatively charged, and when loaded onto the gel and placed in a electric field, it will migrate towards the anode. After the visualization by staining, the size of proteins can be calculated by comparing its migration distance with the help of molecular weight ladder, that is the marker unknown sample. Next, coming to the applications of SDS page. It is used to measure the molecular weight of molecules and used to estimate the size of proteins, and used in peptide mapping, and used to compare the polypeptide composition of different structures, and it's used to estimate the purity of proteins. And sodium dicyl sulfate is used in Western blotting techniques, and it's used in HIV test to separate the HIV proteins. It's used to analyzing the size and number of polypeptide subunits, and it's used to analyze the post-translational modifications. Okay, this is the electrophoresis technique. Serum proteins electrophoresis by agarose gel and sodium dicyl sulfate polyacrylamide gel electrophoresis.